Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss oxidative stress. In the last video, we talked about cell adaptation, cell injury, as well as cell death. One of the most common causes of cell injury is oxidative stress to the cell. So what is oxidative stress? Oxidative stress refers to the damage to cells caused by excess free radicals. There are many different types of free radicals, but especially free radicals that are derived from oxygen, what we call reactive oxygen species, or ROS. So why are oxygen species the most important among all the radicals that one can actually form? The reason is because, as you know, oxygen is taken into the body 24/7 all the time in order to generate energy. So most of these free radicals that cause stress to the cell, they are derived from oxygen. So that's why we call this oxidative stress. Recall from your chemistry that a radical, which in biology and medicine we often call a free radical. A free radical is basically an atom or group of atoms with one or more unpaired electrons. Radicals are unstable and very reactive. They will react with any chemical nearby. So in the cell, if there is lipids nearby the radical, the lipids will react with the radical. If there's a protein nearby, that would react with the radical. And if there's, let's say, DNA nearby, then the DNA would react with the radical. Now, a serious problem with this reaction is that when the radical reacts with some target molecule, let's say a lipid, the lipid itself then becomes a radical. And this thereby propagates the chain of radical formation and enhances or continues the damage to the cell. Although there are many radicals that may form in cells in our body, there are some that are more common. These are the ones that are typically implicated in cell damage, and these include the superoxide radical, hydroxyl radical, hydrogen peroxide, organic radicals, hypochlorous acid, nitric oxide, as well as reactive nitrogen oxygen species such as peroxynitrite. Let's first look at how are free radicals produced in our cells. Recall from metabolism that oxygen is used in the electron transport chain at complex 4 of the electron transport chain, and that oxygen basically becomes water that gives off a lot of redox energy, which is then used by the enzyme ATP synthase to generate ATP. It is estimated that about 95% of all the oxygen that comes to the mitochondria will go through this process efficiently, generating water. However, this process of electron transport chain oxidative phosphorylation is not perfect. About 4 to 5% of the oxygen actually produces superoxide, and that happens, for example, at the coenzyme Q. Coenzyme Q may accidentally transfer an electron to oxygen, and that will form superoxide. So out of 100 molecules that go through this ETCOP, about 4 to 5 will become this free radical called superoxide. So this means free radical formation is part of energy generation. Now, superoxide, they cannot diffuse far from their site of production. However, they can initiate production of other reactive oxygen species. So you can see how a superoxide can lead to the formation of hydrogen peroxide, which can then cause the formation of the hydroxyl radical. So besides metabolism, free radicals can be generated by radiation, ultraviolet light, X-rays, as well as other strong radiations can break water into the hydroxyl radical and the hydrogen radical. This actually is the mechanism of radiation therapy for cancer, because that's what happens. By radiating the cancer cells, free radicals are produced in the cancer cells, and these free radicals then will hopefully kill the cancer cells. So as a side note, it's important to remember that patients on radiation therapy should be cautious in consuming large amounts of antioxidants. We'll talk about antioxidants later, but antioxidants basically neutralize radicals. So they should not take too many antioxidants, because it's going to neutralize the radicals and thereby negate the effects of the radiation therapy. Another way by which radicals are formed in the body is through enzymatic reactions. There are several enzymes, examples oxidases, peroxidases, as well as oxygenases, which we have talked about under metabolism. These enzymes use oxygen as a substrate, and in the mechanism of these enzymes, they can transfer a single electron to oxygen, especially by the metal cofactor, and generate RS. Here's an example of the enzyme xanthine oxidase that converts hypoxanthine into uric acid, and you can see in the process, in this reaction, superoxide is formed. Another example of radical formation by an enzyme is seen in the liver enzyme cytochrome P450. Now, these family of enzymes break down various foreign chemicals, what we call xenobiotics, such as drugs, alcohol, pollutants, and other chemicals. And in the process, they may release free radical intermediates. A good example of this is carbon tetrachloride. If someone is exposed to carbon tetrachloride, CCl4, then the liver cytochrome P450 will convert this carbon tetrachloride into the trichloromethyl radical, as well as the trichloromethyl peroxyl radical. And this is done by the cytochrome P450, especially CYP2E1, that catalyzes this reaction, forming these very strong radicals, free radicals. And of course, these free radicals then go on to damage anything nearby, including lipids, DNA, proteins, and so on. And that leads to fatty degeneration in the liver, fibrosis, as well as cell death of the liver cells by necrosis. There are also several enzymes that can generate hydrogen peroxide, for example, oxidases. So in the mitochondria of certain neurons, there's an enzyme called monoamine oxidase, MAO, and these monoamine oxidases react with monoamines such as serotonin and dopamine, and results in the production of hydrogen peroxide. Now, H2O2 is a stable chemical, it can readily cross biological membranes. So that's the dangerous thing about H2O2. And we'll see later H2O2 eventually can form free radicals. Another example of this H2O2 formation we saw in video 5.25, where we discussed the beta oxidation of very long chain fatty acids, VLCFA, that happens in peroxisomes, and these enzymes produce hydrogen peroxide. So that's another very dangerous chemical that can eventually lead to formation of free radicals. Now, another way that leads to RS production in the body is by metals, especially transition metals such as iron and copper. Now, normally these metals are bound to proteins. Okay, so the metals are bound to proteins such as enzymes, hemoglobin, as you know, and myoglobin has iron, and other proteins. So these metals are not free. They are tightly bound to various proteins, and that keeps them safe. However, if they become free, for example, these proteins break down and the metals are released, now these metals can then react with hydrogen peroxide to produce free radicals. For example, the ferrous metal, the ferrous iron, can react with hydrogen peroxide to produce the very dangerous hydroxyl radical, and this is called the Fenton reaction. As a side note, there is another reaction called Haber-Weiss reaction. In this case, the hydrogen peroxide reacts with superoxide, forming this very dangerous hydroxyl radical. Okay, so this is one of the most dangerous radicals for cells that is implicated in a lot of diseases. Now, another type of radical that may be formed is what we call reactive nitrogen-oxygen species, or RNOS. Many of our cells, such as neurons, endothelial cells, and immune cells, have an enzyme called nitric oxide synthase, or NOS. Now, this enzyme, nitric oxide synthase, can catalyze the formation of nitric oxide from arginine and oxygen. At appropriate concentrations, at low concentrations, nitric oxide serves as a hormone as well as a neurotransmitter. Now, nitric oxide is a gas, so it very easily diffuses through membranes as well as water. Nitric oxide can bind to the enzyme guanylyl cyclase and activate signal transduction cascade. In case of neurons, nitric oxide works as a neurotransmitter, while in endothelial cells, it works as a hormone. However, in immune cells, NOS induction results in large amounts of nitric oxide. These large amounts of nitric oxide that can be produced in immune cells, it can react with oxygen as well as other chemical species to produce all kinds of RNOS, reactive nitrogen oxygen species, such as the nitronium ion, nitrogen dioxide, as well as nitrogen trioxide. And you see that happening here, reaction of nitric oxide with oxygen as well as other chemical species. This RNOS that is produced in very large amounts in immune cells is normally used to destroy the engulfed microbe or whatever cellular debris that the immune cells has phagocytosed. However, when the inflammation is prolonged, what we say chronic inflammation, Then these RNOS at high concentrations present in the immune cells, they can leak out of the immune cells and then cause damage to any cells nearby. So we can see widespread damage to normal tissue that is surrounding the disease site. So this is one of the ways by which chronic inflammation can be very dangerous to our body. Since we are talking about immune cells, let's look at this ROS production in these phagocytic cells a little bit more. So ROS production during phagocytosis is a very important process. Let's see how this happens and what it does. So in the first step, the microbe or whatever cellular debris that is shown here, this will bind to special receptors on the immune cells. So there is the binding step that happens first. And this binding is even more efficient if these immune cells are coated by antibodies, or the cellular debris is coated by antibodies. Once the binding is done, once the microbe binds to the immune cell, then the macrophage or the neutrophil, whatever this immune cell is, mostly in the macrophages and neutrophils, they're very good in phagocytosis. They will surround the microbe by forming these cytoplasmic extensions that we call pseudopods. So this is the step that we call engulfment. In the next step, the plasma membrane pinches off to encapsulate the microbe or the cellular debris. This forms vesicles. These vesicles are called phagosomes. So here we see a vesicle with the microbe or the cellular debris inside the vesicle. These phagosomes will then fuse with lysosomes, forming what we call phagolysosomes. Now in the next step, there's this enzyme called NADPH oxidase that's present on the membrane of the phagolysosome. And this enzyme basically forms superoxide from oxygen and NADPH. So we see here a lot of superoxide that is formed from oxygen. So at this point, there's a lot of oxygen that is consumed by these cells. A lot of oxygen is used up. And we actually call this the oxygen burst. Okay, so this dramatic increase in oxygen consumption by the cells during phagocytosis is called oxygen burst. In the next step, the enzyme superoxide dismutase or SOD will convert this superoxide into hydrogen peroxide, H2O2. So while this is happening, H2O2 is being formed in the phagolysosome. The phagocyte, the cell, will also transport the cytoplasmic granules rich in the heme peroxidase enzyme called myeloperoxidase, MPO. So MPO is transported from the cytoplasm into the phagolysosome cavity. Now MPO will then catalyze the reaction of hydrogen peroxide with chloride, and that will lead to the formation of this HOCl, which is hypochlorous acid, HOCl. This is a very powerful oxidant, and this HOCl will help degrade the bacteria or whatever that has been engulfed, whether it's cellular debris or it's some sort of microbe, will start breaking down because of this very powerful HOCl. So besides HOCl, the lysosome itself, because remember this is the lysosome has spilled all its contents into this phagolysosome. So there's a lot of lysosomal enzymes also present in this phagolysosome. And these lysosomal enzymes will also help break down the ingested material into chemicals. In addition, the hydrogen peroxide will react with ferrous by the Fenton reaction to produce hydroxy radicals. So not only do we get HOCl, lysosomal enzymes, now we also have this very powerful hydroxy radical that will help break down the ingested material. Now, the superoxide formed here can also react with nitric oxide, and this will lead to the formation of this reactive nitrogen oxygen species, RNOS, and this RNOS will also help break down the ingested material. So what we have in the phagolysosome is these lysosomal enzymes, hypochlorous acid, the hydroxyl radical, and then this RNOS, and all of these will help break down the ingested material, whether it's a microbe or it's some sort of cellular debris. So some of these possible outcomes of this phagocytosis will include the following. One, the powerful free radicals that are produced in the phagolysosome will lead to the complete degradation of the ingested material. Another possible outcome, there could be leakage of free radicals from the phagocytic cells. So these free radicals might leak out of these cells, and that will damage any surrounding normal tissue. And this is a problem with chronic inflammation. Another outcome is that the extracellular release of free radicals may increase the expression of cytokines and other inflammatory chemicals, and that will amplify the inflammatory process. So we have seen several different ways by which free radicals can be generated in our cells. So now let's look at how do these excess free radicals attack our cells. What do they actually do? So theoretically, free radical is so reactive that it can attack and damage any biomolecule nearby. However, there are three important aspects that we have to look into when we look at cell injury, and this is how these free radicals attack membranes, how these free radicals attack proteins, and the attack on DNA. Now, membranes have a lot of lipids, and if you remember from the earlier videos, several of these lipids are unsaturated, they have double bonds, and these double bonds are especially susceptible to free radical attack. So any membrane, plasma membrane, but especially the mitochondrial and lysosomal membrane, if the plasma membrane is attacked, that will cause leak out of cellular constituents. Mitochondrial membrane, as you know, mitochondria is very important for energy generation. Lysosomal membrane may leak out all kinds of very powerful enzymes. So when these free radicals attack these membrane lipids, they form reactive peroxides. Now these peroxides then will go attack more lipids, and that will lead to an autocatalytic chain that results in more and more damage to the various membranes in the cell. Now, free radicals attack on proteins can cause cross-linking by disulfides. The sulfhydryl group is especially very reactive and can react with free radicals. Remember cysteine, and can react with another acids group, and that two acids will form disulfides, SS, and that will lead to protein cross-linking. Of course, proteins per polypeptide chain can also be broken down by free radicals into fragments. And free radicals can affect proteins such that the proteins cannot fold properly. Okay, protein folding is affected, so that leads to misfolded proteins, proteins that are not folded correctly. And this presence, this accumulation of misfolded proteins in the cell, especially in the endoplasmic reticulum, triggers a response called the unfolded protein response, UPR, unfolded protein response. You will hear this word quite often in pathology. And UPR, the end result of UPR is first to try to decrease protein synthesis so that the cell doesn't keep on producing misfolded proteins. But if that doesn't work, then the cell will die by apoptosis. Now, if the free radical attacks DNA, that can lead to mutations as well as DNA breaking into fragments, and this has been implicated in apoptosis, in aging, as well as in cancer. So there are several different ways by which our cells protect themselves against excess free radicals. So let's look at these different mechanisms, how we protect ourselves against free radicals. So one is the enzyme we talked about earlier, superoxide dismutase, or SOD. This is found in the cytoplasm, mitochondria, and also sometimes secreted outside the cell. And basically what it does, it will eliminate or get rid of the superoxide anion, convert it into H2O2. However, remember this H2O2 is a dangerous chemical too. So this H2O2 that is produced here or elsewhere must be rapidly destroyed because it very readily diffuses across membranes and then react with metals such as iron to form a more dangerous hydroxyl radical by the Fenton reaction, or it may react with superoxide to form the hydroxyl radical by Haber-Weiss reaction that we talked about earlier in phagocytosis. So for this we have another enzyme called catalase. Catalase destroys hydrogen peroxide into water and oxygen. Now catalase is one of the most efficient enzymes in our body. If you remember turnover number that we talked about in enzyme kinetics, how many substrate molecules are converted into products per second, the kcat, the turnover number for catalase, is estimated to be around 3 million per second. That means 3 million hydrogen peroxide molecules can be converted to water and oxygen per second. Very, very efficient enzyme, and you can see why this is so important to protect our cells, especially to protect against this hydrogen peroxide. So besides catalase, we do have another enzyme called glutathione peroxidase. This also breaks down hydrogen peroxide into water. In order to catalyze this reaction, glutathione peroxidase requires this tripeptide called glutathione, which we abbreviate as GSH, and the structure of GSH is shown here. Notice it's made up of 3 amino acids: glycine, cysteine, and glutamic acid. So notice this is the alpha carbon of glycine, and then we have the alpha carbon of cysteine, and here we have the alpha carbon of glutamic acid. So the amine of glycine, the alpha amine of glycine, you can see is connected to the carboxyl of cysteine by a normal peptide bond. However, the combination, the joining of cysteine and glutamic acid is different. You can see this carboxyl group is not the alpha carboxyl group. The alpha carboxyl group is here. So this is the gamma carboxyl group of glutamic acid that is joined to the alpha amine of cysteine. So this is a little bit of a different linkage here. The important group here we want to look at is the sulfhydryl. So basically this whole thing here we abbreviate as G, and then the acids we write acids. So this is glutathione, the reduced form of glutathione, or GSH. So in this glutathione peroxidase reaction, GSH will react with hydrogen peroxide here. You produce water, and the GSH will be converted into GSSG. Basically, two of these GSH come together to form a disulfide, forming GSSG. This is the oxidized form of glutathione. So glutathione peroxidase, which is very important to destroy hydrogen peroxide, this enzyme, which is a selenoprotein, it requires selenium to function. That's why sometimes we say selenium is, and sometimes we consider selenium as an antioxidant, because selenium is necessary for glutathione peroxidase to destroy hydrogen peroxide, which can produce very powerful free radicals into safe chemical water. So as mentioned earlier, this glutathione after the reaction will become, this reduced glutathione, GSH, will become oxidized glutathione, GSSG. So in order to make sure that we don't run out of GSH, because GSH is becoming GSSG every time it reacts with hydrogen peroxide, we need to make sure that this GSSG, this oxidized glutathione, can be reconverted back to GSH so that the enzyme can use GSH to destroy another molecule of hydrogen peroxide. The conversion of oxidized glutathione back into the useful glutathione, the reduced GSH, is carried out by another enzyme called glutathione reductase. This uses NADPH. So basically NADPH is used to reduce the oxidized glutathione into reduced glutathione. The NADPH in turn will become NADP. Most of this NADPH required for this reaction by glutathione reductase comes from the pentose phosphate pathway that we talked about earlier under metabolism. And remember, pentose phosphate pathway, also called the hexose monophosphate shunt, or HMS, is a pathway where glucose is converted to ribose and other sugars. One of the important functions of this pathway is to produce NADPH. And you can see now one important use of NADPH is to help regenerate reduced glutathione so that glutathione peroxidase can keep on eliminating excess hydrogen peroxide. So this molecule glutathione is a very important chemical. It's a very good antioxidant because the sulfhydryl group is very reactive, can react with free radicals. The liver uses a lot of glutathione for detoxification of drugs as well as other xenobiotics, other foreign chemicals. A few words about acetaminophen poisoning. Commonly, this drug is called Tylenol. So in Tylenol poisoning, what happens is that the liver cytochrome P450, especially the 2E1, this cytochrome converts about 10% of the Tylenol that is ingested into a very highly reactive drug metabolite known as N-acetyl para-benzoquinoneimine, or NAPQI. Now, MAPQI is very reactive. It will attack all kinds of proteins, and especially if the mitochondrial proteins are attacked, that is going to lead to oxidative stress. And then that oxidative stress is going to cause injury to the liver cells. Now, this NAPQI is inactivated into a safe compound by reaction or by conjugation with this glutathione. So acetaminophen poisoning, Tylenol poisoning can lead to serious liver injury. And the treatment for acetaminophen poisoning is administering this chemical here called N-acetylcysteine. NAC, which is taken up by the liver cells, will help restore the concentration of GSH. Now, we cannot give GSH directly because GSH is not taken up. Glutathione is not taken up by liver cells. So NAC is the treatment for acetaminophen poisoning. Now, another way we can protect ourselves against free radicals are using antioxidants, free radical scavengers. That includes vitamin E, vitamin C, carotenoids, as well as flavonoids that are present in the diet, especially fruits and vegetables. And there are also other small molecules that can act as antioxidants, example, uric acid, several low molecular weight thiols, some proteins, especially rich in cysteine, as well as melatonin that's produced in the body. So how does these antioxidants work? One typical mechanism how this works is, let's look at ascorbic acid, for example, vitamin C, ascorbic acid. Basically, the ascorbic acid will donate a hydrogen atom to the free radical. So the F dot will take the hydrogen and become FH. So thereby neutralizing the free radical. Now the vitamin C may itself become a radical, in this case ascorbic radical. Okay, so the antioxidant may become a free radical. But if you look at the structure of these antioxidants, that free radical can be stabilized by resonance stabilization. So although the antioxidant is a free radical, it's not a reactive free radical because it is a stable chemical. Stable chemicals are not reactive. So because of the chemical structure of these antioxidants, which can be, the radical form can be stabilized by resonance stabilization, these antioxidant free radicals are not reactive, they are safe. So that's one way by which some of these antioxidants help neutralize free radicals. Now, while inside cells we have this very robust antioxidant defense system, including SOD, glutathione, peroxidase, catalase, outside cells, like in the plasma, there is very limited AO defense. So there is no catalase present outside cells, negligible amounts, very small amounts of superoxide, dismutase, or glutathione peroxidase present in extracellular fluids. So what are the major antioxidant defenses in the, for example, in the bloodstream? Albumin, which has quite a few thiol groups. The antioxidants we take in our diet, for example, vitamin C and the flavonoids and so on, and some chemicals present in the blood such as bilirubin and uric acid that come from breakdown. Bilirubin comes from the breakdown of heme. Uric acid, as we discussed in an early video, is formed from the breakdown of purine nucleotides. They can also act as weak antioxidants. Now, one very important disease, cardiovascular disease, due to a blockage of blood vessels, what we call atherosclerosis. One of the important events that happens in atherosclerosis early on is the oxidation of LDL by reactive oxygen species. Okay, so this oxidation is a very important factor for the progression of atherosclerosis. So clearly we must try to reduce the ROS in our bloodstream. And this is one of the main reasons why physicians, nutritionists tell patients, tell everyone, eat your fruits and vegetables every day, because you need these dietary antioxidants every day to help the albumin as well as other chemicals take care of this ROS that is being produced all the time in the body. Okay, so that's all about free radicals, a very, very important topic that you will come across in many diseases in the body. So whether you are a healthcare professional or a biomedical researcher, ROS is one of the words you will hear very, very often. Okay, so that's the reason why we have a longer video this time. Extremely important concept that you need to understand and keep in mind as you go through your professional training and your career. All right, in the next video, we shall look at programmed cell death. Until then, once again, thank you for supporting this channel. Have a wonderful, wonderful day.